Welcome to the Art Biolab channel. Have you ever stopped to wonder if it's possible to grow an entire thriving plant, not from a seed, not from a cutting, but from just a tiny handful of cells? Well, it might sound like something straight out of science fiction, but this is the reality of a remarkable science called plant tissue culture, and it's happening in laboratories all over the world every single day. So let's explore this fascinating world together.
So, here's our roadmap for this journey. We're going to start with the fundamental science, the biological miracle that underpins this entire field. Then we'll look at how it's completely changing the game in agriculture and conservation. After that, we'll take a peek inside the lab to see the actual process in action. And finally, we'll wrap up with a balanced look at the very real challenges and considerations.
All right, first things first. We have to get to the very heart of the matter. What is the core principle in biology that makes it possible for a nearly invisible piece of a plant to grow into a whole new organism?
Well, at its most basic, plant tissue culture is really about creating the perfect, exclusive environment for plant cells to thrive. Think of it like a five-star all-inclusive resort. It's a totally sterile, nutrient-packed medium where a tiny piece of a plant gets absolutely everything it could possibly need to grow, all tucked away safely inside a flask.
Now, the scientific concept that makes this whole thing possible is a remarkable property called totipotency. And this really is the magic right here. It's the idea that every single living plant cell holds the complete genetic blueprint, the entire instruction manual to build the whole plant from scratch. The roots, the stem, the leaves, the flowers, it's all there coded inside one cell.
And you know, while this technology feels incredibly modern, its intellectual roots stretch way back. This isn't some newfangled idea. It's built upon the foundational cell theory from the 1830s. The dream itself was first articulated by Gottlieb Haberland back in 1902. And after decades of work, scientists finally achieved sustained cultures in the late 1930s, paving the way for the explosion in its use we've seen since the 70s.
Okay, so that's the science. It's fascinating stuff. But how does this elegant laboratory technique translate into a technology that has a massive real-world impact? I mean, why does this matter so much?
Well, the applications are frankly revolutionary. We're talking about the ability to produce millions of genetically identical clones from one superior plant. We can conserve critically endangered species, create a perfectly reliable supply of medicinal compounds, and even produce crops year-round, completely independent of the seasons.
And this right here is one of the most powerful uses. Just imagine a priceless variety of a plant, maybe a vital food crop, but it's riddled with a virus. Well, by taking an impossibly small sample from the very growing tip, the meristem, an area that viruses often haven't reached yet, we can regenerate a completely clean, healthy version of that exact same plant. It is, for all intents and purposes, a biological reset button.
So what does it actually take to pull all of this off? Well, as you might imagine, it all comes down to creating an environment that is meticulously controlled and, more importantly than anything else, absolutely unquestionably sterile.
Let me just say this as clearly as I can. In the world of tissue culture, contamination is the ultimate enemy. A single stray bacterium, one invisible fungal spore floating in the air, if it gets into your culture, it will multiply with astonishing speed, stealing nutrients and killing your plant tissue. So sterility isn't just a guideline, it is the absolute non-negotiable law of the lab.
And to achieve that level of clinical cleanliness, you need some very specific equipment. You need an autoclave, which is basically an industrial-strength pressure cooker to sterilize media and tools with superheated steam. All the delicate work happens inside a laminar flow hood, which creates a curtain of ultra-filtered air to keep any contaminants away. And then, of course, you have incubators that provide the perfect controlled climate for the plants to grow.
All right, so we understand the science, we've seen the sterile laboratory. Now let's walk through the actual process. What is the step-by-step journey that takes a tiny piece of tissue and transforms it into a living, breathing plantlet?
It truly is a delicate and methodical art. First, you carefully select the best piece of starting tissue. That's your explant. Next, that explant gets a very harsh bath in sterilizing agents like bleach to clean its surface. Then, it's carefully placed or inoculated onto that nutrient gel. From there, it goes into the incubator to multiply and regenerate new shoots. And finally, once you have tiny plantlets, they have to be slowly and carefully acclimatized or hardened off to prepare them for life in the outside world.
Now, for all the amazing things this technology can do, it's certainly not a simple magic bullet. It is an intricate, often expensive process that demands a tremendous amount of skill and precision. So it's time for a bit of a reality check. With all of this incredible potential, you have to ask, what are the real-world hurdles? What are the limitations that prevent this from being the standard method for growing every single plant everywhere? This really lays out the core trade-offs quite well. On the one hand, yes, you get incredible benefits like mass cloning and disease-free plants, but on the other hand, the setup is expensive and the process is extremely labor-intensive. It demands a very high level of technical skill, and as we've already seen, one tiny mistake can result in a total loss. On top of that, growth can sometimes be slow, and in some cases, long-term cultures can even lead to unexpected genetic mutations.
And let's put a number to that risk of contamination we keep talking about. The stakes are just incredibly high. A minor breach in that sterile protocol doesn't just affect one little petri dish. A single airborne contaminant has the potential to spread and cause the failure of over 90% of all the cultures in a lab. It is a constant, high-stakes battle that requires unwavering precision.
And this brings us to a final, larger question to think about. As our ability to create and cultivate plants entirely within the controlled environment of a laboratory becomes more and more refined, what does this ultimately mean for nature itself? How will this profound power to replicate and regenerate life on demand continue to shape our agriculture, our conservation efforts, and perhaps even our very relationship with the natural world?